Welcome to the Conversational Flow, a KCAS bio podcast.
Hello, and welcome to the 23rd episode of the Conversational Flow, an official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. Unlike KCAS' other podcasts, the Weekly Bioanalysis, which features news and topics more generally associated with the world of bioanalysis, the Conversational Flow focuses more on the niche of flow cytometry and its role within the greater industry. My name is Adam, and I've been in the flow cytometry space for over 15 years. I'm here with my co-host, Brian.
Thanks, Adam. My name is Brian. I've been in the flow cytometry space for over 14 years.
Brian and I are members of the growing leadership and scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. While our KCAS headquarters are located in Kansas with additional locations in Europe and a partner location in Australia, we're both here in Philadelphia, and Jeremy, our producer, is in Missouri. So we're truly all over the map. We're thrilled to have you listening to our 23rd episode of our podcast, which will be available virtually everywhere. Wherever you choose to find and play your podcast, you'll likely find the Conversational Flow. We welcome all of you, whether you're joining us for the first time or if you're a regular listener to any of our other podcasts. Today's episode will be a review of the latest news and resources and then a focus on a topic of our choosing before discussing any feedback we've received from you. We're constantly looking for topics, and we'd be happy to discuss something for you that you want us to cover. So again, we're thrilled to have you here, and we're looking forward to a fun episode today. To kick us off for episode 23, Brian is going to go over news and resources before diving into today's topic. Brian?
Thanks, Adam. We've got an especially crunchy agenda, actually, today. And I say crunchy just because we've got a couple things sort of off the beaten path for our normal podcast fare. So the first up is some medical research news. And the first article I brought up today is Breakthrough Lung Cancer Treatment Supercharges Immune Cells with Mitochondria.
Yeah, this is really cool. I hadn't heard about this before, and diving into it, really looking at what's going on. You transplant mitochondria into the tumor space, right? And that will actually engage the immune cells. Now, the thing that I was kind of curious about was not having known much about this beforehand. What do you just like take mitochondria out and then you have to inject it into cells? Like that seems so impractical and so much work, very laborious. And no, it's not that. I mean, essentially injecting it into the area, the mitochondria are much more fluid in between cells than I had realized before reading this article and then reading support information.
That's exactly right. And that was my first thought as well, that who is taking all these immune cells and carefully injecting like 2 mitochondria into each one and then going on to the next. But you are correct. It is a much more robust procedure than that. As you say, it's still a relatively difficult one, which is why I mentioned that it's sort of crunchy and off the beaten path. But I found it to be an interesting exercise in terms of what could help with response to cancer. And if they're looking at the metabolic requirements of immune cells and they're exploring that through actual mitochondria transplantation, it becomes a much more sort of wider and generalized application of investigation here. So yeah, it was interesting.
Yeah, and you read about the cancer cells actually impacting the functional performance of the mitochondria of the immune cells that are already there. So adding in mitochondria from another source helps to reinvigorate these cells, right? Everyone knows mitochondria, the powerhouse of the cell, right? And so here, I think the thought now going through my head is, obviously scaling to be able to get enough mitochondria for such a platform, you're dealing with many of the same things as any cell therapy platform. And so it'd be interesting to see how this can then grow and scale, if it would be inhibited in the same ways that other cell therapies, for example, would be.
You're exactly right. And it could become, if this were translated into a clinical treatment directly, you're absolutely right that this could be a very expensive and or very risky type procedure that would be limited to pretty severe diseases.
Hmm.
But I think just the basic science of it and the mechanism of it is what could be relatively widely applicable there. So a good basic science research that we don't always get to review. So yeah, that was my first article. The second one is more in the clinical space in terms of immune dodging cells giving diabetes a treatment shot in the arm. So that is the um, marketing ready headline they gave there.
Well, it actually is 17 shots in the arm, I think it was. The one patient that they had tried it on had 17 injections of these genetically altered pancreatic cells. Now, you think genetically altered, I mean, this is really going along the allogeneic pathway that we're constantly talking about with cell therapy. And so they removed a couple of genes, HLA1 and 2, to allow the cells to not be destroyed by the immune system. However, if they don't also then increase CD47, then the cells would still be destroyed. So the three main modifications they'd done: injected 80 million cells, which evidently was about 7% of a repair dose. So not a cure, was actually producing insulin, however, not to the extent that was needed to be a cure at the time. So further investigation is occurring.
That's it. So it's interesting from a couple perspectives. Definitely, the stealth cell part is the most relevant to flow cytometry techniques, but certainly diabetes is a huge field overall, and having a one-time treatment that could do something would be much better. Probably not a 17-time treatment, but a one-time treatment, I think we could get behind. So
Yeah, that was the thing that they led with too in the article. And so I just the juxtaposition there of the title saying "a shot in the arm."
yeah,
But yeah, if it works, and this was 80 million cells, 7% repair dose. If it works, I'm sure that even if it is 17 injections, that means that you don't have to manage with insulin the rest of your life. And so would definitely be beneficial.
yeah. But the stealth cell modifications that you mentioned are always critical. In this one, these are, I guess, tissue resident cells. So ideally, not as exposed to as many immune agents as they otherwise might be, but certainly with a lot of the CAR T and various CAR treatments that we talk about a lot on this podcast, the cell becomes absolutely critical to make sure that these treatments are long lasting or long lasting enough to be effective.
And to your point, I was surprised they analyzed this 3 months after the injection, and they had stayed within the same area. So really, it was a cool thing to note there and understand how it just continued to work. To your point.
That's it. All right. So moving on to investment news. The first article I've got up is a Novartis investment in Matchpoint Therapeutics for anti-inflammatory therapies.
And so Matchpoint focuses specifically on covalent binding therapies, essentially the strong bond, right? The really making sure that therapy stays where it should, rather than binding and then getting knocked off. And so here, using that technology, and then what they had mentioned was specifically targeting a transcription factor that's key for a number of inflammatory conditions. I didn't get to seeing what specifically the transcription factor was. Did you, Brian?
I did not know. And I think that they have this based on their lead candidate, but they do have a pipeline of other things that Novartis is interested in, which is why the size of this deal got to where it was. So that's a $1 billion, they call it biobucks for the most part deal, which will have some upfront, but most of it based on how successful this or other therapies in the pipeline would be.
Yeah, and you're talking about interest and activity. I know at the beginning of the year, one of our podcasters said that there wasn't too much in M&A, right? I think ever since we did that podcast has been a lot. And specifically here with Novartis, they have the 4-year alliance with Profound for $750 million in the area of cardiovascular therapy. And then they also have the acquisition of Anthos, specifically with eyes on Kyorin, which is a pre-inflammatory drug. So they've been very active this year.
Okay. Well, in that case, we'll help the economy out and say that, oh, we don't expect any more M&A for this year. So
Absolutely. There we go. I love it.
yeah, wonderful. And then the second investment news I had was an interesting take on things where instead of acquiring companies, Bristol Myers, so BMS, and is partnering with Bain Capital to spin out an immunology biotech. They're called a shell company, essentially.
Did you see what they're referring to this company as?
I saw it as Nuco for a while.
Newco, short new company.
Yeah, right. I mean,
I love that. I mean, if you can't have a little bit of fun, what can you do? Yeah, so they're allowing focus on 5 experimental immunology therapies. 3 of them are clinical stage, 2 phase 1 ready programs with targets including TLR7 and 8, TYK2, IL2, IL18, and IL10. And so of the ones that are clinical stage, we've got afimatorin, which is the TLR7/8 inhibitor, and then BMS 986322, which is an oral TYK2 inhibitor for psoriasis. And so, yeah, it looks like basically it's kind of shifting focus where within the larger umbrella of BMS, they're having one focus, right? Whereas then they're parsing out other therapies, other modalities outside that, they may not necessarily prioritize within the broader umbrella. So they want to make sure that they still happen. And BMS comes out of this, I think, with 20% essentially from this. And so here we talked about Novartis, right? BMS had reached an agreement with BioNTech earlier in June for BNT 327 to co-develop and co-commercialize, and then also acquired 270 Bio for $286 million, which is cell therapy, in March. So again, a lot more M&A activity from where we were in the beginning of the year.
it is. And, for a lot of those acquisitions, that's pretty standard fare for BMS, right? Where they go in and buy a company or an asset and commercialize it, and that's a standard thing. This specific news article, in terms of forming this immunology biotech shell, is very similar to how companies like the various Vants, they call them, so Roivant and Spiravant and Immunovant, sort of operate, where they allow the risk to be sort of parsed out to different drug products as opposed to containing all of them internally. So it makes them at least look more capital efficient, because more of the core BMS bottom line is not going to R&D, it's going to commercialization, and allows these smaller, more nimble companies to take the larger risks there. So
it's amazing too, just looking at the larger companies, how you just have this migration in and out of companies and assets, right? Under the umbrella of like a BMS and Novartis or whatnot, where they're purchasing companies, they're signing deals, but then they're spinning off companies. And it's just like this conveyor belt of science and therapies. It's very fascinating to watch. Now, I do have a really serious question for you, Brian. The people who work at Nuco, what do you think they refer to BMS as?
I feel like do they refer to it as Old Co? Are you saying? Or
yeah, there you go.
No.
Sorry, I amuse myself, and I really have to stop that sometimes.
No worries. Most of the time they just call it the mothership, right? So
Yeah.
I don't know. We'll have to ask them at some point.
Yeah, Old Co. There we go. That's good.
Yeah. So exciting times there. And speaking of exciting times, of course, we've got some exciting news at KCAS because our Lyon site has implemented the new LISA platform.
Yeah, so why don't you talk about New LISA? What is the change here? What is the important takeaway people should have from this?
So New LISA is a really, I would say, advanced new way to analyze the quantity of proteins present in a solution. So it can get to higher levels of sensitivity or be used to multiplex a lot of different measurements all in one. And it does that by incorporating a different type of probe, which can be read out by PCR or PCR analysis, as opposed to a standard ELISA type format where you're relying on other proteins to sort of directly tag that, if that makes sense.
So we're not just talking like a giant spectrophotometer that's giving us the OD 260, 280 and, leaving it at that. We're going a whole lot more deeper than that.
We are. So there's an amplification step in there, right? Which is what helps to give it that extra level of sensitivity.
Mm-hmm.
And also because it's PCR based, it allows PCR or NGS, I guess I should say, it allows the multiplexing, because as long as you've got separate probes and primer sequences, then you're unlimited in terms of the number of different analytes you can detect at once.
Mm-hmm.
So
Nice. And
yeah.
so this machine, remind me, you mentioned about it being in Lyon. Is this also in Olathe?
Right now it's Lyon only, but we're of course evaluating how quickly sponsors are adopting it and seeing if we need to stand it up in more places.
Mm-hmm. Excellent.
Yeah. All right. So that is all of our exciting news for the month. And in terms of our paper and our main topic, you picked out this one, Adam.
Yeah, so we talk about a lot of common topics. I think cell therapies, a lot of very pertinent, very burning topics that we've got going on all the time. Immunophenotyping, receptor occupancy assays, harmonization, you name it, validation. Kind of wanted to go a little bit different just because the core company, right, from the olden days, always specialized in making a la carte analyses, whether it was panels, whether it was different matrices, so on and so forth. And so I kind of wanted to get back to that a little bit, pining for the old days in that respect, I guess. And so the paper is Sputum Analysis by Flow Cytometry, an effective platform to analyze the lung environment. And this is published in +1, impact factor of 2.6. Lead author was Lydia Bedurka. And so really looking at a different sample type than what we get on the daily, right? I mean, every day we're getting whole blood, we're getting cytochecks tubes, we're PBMCs, things of that nature. And, sputum, occasionally we get requests for something like this. And so really kind of wanted to dive into what people are looking at. Now, what is the allure of doing something like a sputum? Well, it's a little bit simpler, obviously, than having to draw blood, right? As you read through the paper, you do realize, though, that it's not easy per se to get a true good sputum sample. And I think that was one of the things that the authors kind of highlighted through. And so the overarching thing here is trying to look at lung cancer and identify lung cancer by the use of sputum, as opposed to some of the other tests like a low-dose CT scan, which is more prevalent, however, suffers from having low specificity. So curious, Brian, what are your thoughts before I continue to ramble on?
It was interesting, right? And it goes along with the standard sort of flow approach of go where your biomarkers are.
Mm-hmm.
So if you can find these biomarkers in this type of sample, then it is a lot nicer for patients, right? You don't have to try to scrape their lungs or get like an actual pathology sample. You don't have to try to look through their rib cage to get an idea of how things are going. You get the closest that you can get to secreted signals that can tell you if things are cancerous or not. And ideally, depending on how this sputum is harvested, it is heavily influenced by the presence or absence of pathology in the lungs. So from that standpoint, it's interesting. But as you say, the the challenges of analyzing sputum are high. Um,
Yes. Now, but I mean, if you're looking at the prevailing tests, one of the things that they had mentioned was that 96.4% of the positive LDCT scans were false positives in this trial. And that's a lot, right? you know, Basically the way they said, if you go back to your biostats, you look at sensitivity versus specificity. You've got sensitivity being the true positive rate, so you want to minimize false negatives, and then specificity being the true negative rate, so minimizing the false positives. And so essentially here, what you're seeing is that LDCT has a sensitivity of 93.8%, which, that's pretty high. However, the specificity is 73.4%. And so that's where you're ending up getting so many of these false positives, essentially, healthy patients that don't have cancers that are identified as positive.
that's it, and that's where you really have to make sure to screen the population before they get that low specificity test. Otherwise, your healthy population or your non-lung cancer population is always going to be, extremely massive compared to your impacted lung cancer population. So, being able to stop those known negatives before they get to the test is critical. Otherwise, you get a lot of people that get some very, very scary news that needs to be checked beforehand. Um, and this is one of those options to do that.
hmm And now you start diving into the flow cytometry portion of it, right? First off, the way that they separated out, starting out with 210 samples, and then going through reducing it based on the quality of the sample, any technical issues in collection, so on and so forth, and then separating it out between high risk and cancer. So you've got a subset that you already understand what the difference is, and then going through building the panel. So one of the key things that they mentioned here was a viability stain, which we've talked about before. I mean, everything that we run, unless it's already fixed, we're using a viability dye. They did have porphyrin TCPP, which is supposed to aggregate to cancer cells. We'll talk about a little bit as we get further in why that didn't seem to work as effectively as they were going to hope, as they had hoped it would. But then we've got macrophage marker, anti-CD206. We've got a grand marker, CD66B, and then CD3 and CD19 for your T's and B's there. And then finally, they did have a couple of epithelial markers. And it seemed like the most important things that they had identified were the plus or minus CD45 specifically, and then looking at those epithelial markers specifically. Less so was that TCPP labeling. And so go ahead. Sounds like you got something you want to say.
Yeah. So I agree with your analysis. Like, definitely there were quite a few markers, and they were trying to determine what was important and what they needed and what they didn't. I feel like this paper did a good job, and potentially a better job of than others that I've seen, in terms of describing some of the struggle. Right. Like, I felt
Yeah.
like I could see the development pathway, and where they tried to go one direction, ran into a roadblock, had to go another direction, went down that path a bit, found another roadblock, and went down another direction. So yeah, I mean, the struggle is real sometimes as you're looking at
I mean,
these.
sometimes, like, I feel like it's the majority of times you find out which ways you can't go before you can find out which ways you can go. And they did a great job, to your point, of highlighting that. And I think it's great to be out there in the scientific literature because it allows you to start where they stopped, kind of thing. Some of the other things that they noticed, the SEC cells, the squamous epithelial cells, are highly autofluorescent. And so removing that, but interestingly, they were removing that using a viability gate. So the vast majority of these cells were dead. And so you could just remove them that way. They did also identify what the minimum number of cells they felt were too few to be diagnostic by flow, and that was about 1.5 million cells. So in talking about that TCPP and why did it not aggregate more to the cancer cells. They had speculated that the samples were fixed first, and then the next day they had stained with the TCPP, and so perhaps that had impacted in some way the ability for it to properly stain and bind. And so that's where perhaps a modification to this procedure would be to do that before that final fixation step.
That's it. And then also I noticed that they were doing this on either a Fortessa or an Aria.
Uh-huh.
And, anytime you say autofluorescence, I get excited about what does this look like on a Cytac, right? So
Fair, yeah. That was right away when I saw that. I thought the same thing. You get to the opportunity of using autofluorescence extraction there. I wonder what would that do? Is that just as effective as a viability gate here? What would happen if you couldn't use a viability gate for these samples? Say you were going to get cytocheck samples, and you still wanted to remove that. Now you've got that ability with spectral. So does that open up the ability to collect? I mean, obviously here you're collecting sputum, so it's different, and I will apologize, I don't often collect sputum. So is there a certain fixative and like akin to a cytochecks that's used in that respect? But yeah, so very interesting.
yeah. And so just sort of dovetailing with this paper, I made our main agenda around how do we even deal with non-blood matrices in general, right?
Hmm.
And I think a lot of what you mentioned is critical in terms of that start. So from the time that the sample is collected, or even before that, from the time the sample is currently in the patient until the time they get to the lab, how do we minimize variability in each and every one of those steps? This paper was in a diagnostic situation, so it was I'll call it relatively controlled status of not needing to get transported long distances. But of course, with clinical trials as well, we have to worry about potentially 1 or even 2 day shipping timelines, depending on where in the world they are. So how do we make sure that as you collect sputum, you're doing it consistently, that the person collecting it is doing it the same way, uh, that they're not doing something that could lead to a bias overall, and then getting it to the right spot at the right time at the right temperature, and then making sure that anything the lab is doing is the same, because they mentioned quite a struggle with filtration steps as well.
Hmm. Yeah, this sample type was interesting to me because I remember being part of studies back, I want to say 10, 12 years ago at my previous position where they would do exhale breath collections. And
Yeah.
so you would have to breathe for several seconds, whatever, into a tube that essentially was frozen, and it would collect the cells, collect the aspirate, whatever. Yeah, so a little bit different here. They actually utilized an apparatus to collect the sputum more effectively. And I think that is one of the things that they were talking about was kind of identifying an ideal method there and making sure that that's used consistently, because different methods had higher variability in that end result, the quality of the sample.
That's it, right? But even with this method, like they relied on people to basically self-collect, if I'm reading this correct,
Hmm.
and then the samples were collected over potentially several days,
Hmm.
and then shipped to the lab, and then immediately tested.
Yeah.
So compared to our normal draw blood, ship it within 2 to 4 hours, it arrives at the lab next day. Definitely a challenge. And then with this type of sample as well, certainly contamination or bacterial growth becomes a real risk. And I think that's what they found as part of the sample collection there as well.
I mean, when you're talking about sputum, yeah, the bacterial growth, it's already there, right? It's part of what the lungs do is filter out, as you breathe in, and you get that mucus layer that kind of prevents any of that sort of thing. And so now you're expelling it. And so this is where, you're absolutely right. I'd really be curious to see further differentials between the initial collection versus the second and third day. What does that look like? Now, they did mention, I believe, that these were, what was it? It was put on ice, right, over that period of time?
Yeah, they stored it in a refrigerator in
Yeah.
theory.
So you would obviously reduce growth, but as everybody knows who puts anything in a refrigerator, it doesn't stop it. We've all had those examples of leftovers that look pretty disgusting. Usually it takes about a week or so. Hopefully your leftovers are more tasty than that allow you to finish it
Oh,
up before
okay.
then.
All right. we gotta move on. So then once it gets to, so two things, I guess. For oh goodness. For I can't even.
Did you
Um,
just like totally derail? Did I just
I just
derail your
derailed. Oh
train?
yeah, no, it's done. Um, whenever you're taking a sample like that too, then I always wonder, especially compared to like CT imaging or other things, you're losing some of the spatial information that could be important as well. So you're getting fluid from all over the lungs, not just in one spot. So is it a good sampling or is it not? We're talking about sputum specifically, but if you think about urine or eye wash or tissue, and which part of the tissue sample or which part of the skin are you taking a biopsy of or something like that, there start to be a lot of questions. And so it's pre-analytical considerations of how well are you sampling what you need to sample. So how sure can you be that you're taking a representative picture of what is currently there in the person.
Hmm.
So that's definitely critical. And then when you actually get to the laboratory, usually with these types of samples, there's a lot of processing that might be needed. So in this paper, they talked about the filtration and the different steps they use to try to filter out more and more of the process. But each of those steps carries a risk of impacting the sample,
Hmm.
right? Either just impacting just total yield, or more concerning, is it biasing your sample by removing one type of particle that or biomarker that could be critical to understanding the whole picture.
Right. It's always a discussion that we have. If there's a qualified assay, not qualified assay, if there's stickiness in the sample, do you add DNAs? Is it something where you filter? Obviously, as part of our SOPs, we have built-in filtration. Is just that doesn't alter the qualified status. But I mean, these are all considerations. How much does that change the sample, the percentage of different cell types that you have in there, the makeup? And it didn't seem like they felt in this paper specifically that filtration really helped very much. And now I think going and taking all of this a step further, I mean, you alluded to it at the outset, is that multiple um, and analysis paradigms are required for a specific diagnosis here. You've got the initial test that we had talked about. Let me make sure I got my acronym correct. The LDCT, right? And then on top, what they had done was they took the sputum samples here, and then they'd done a biopsy, and so they were able to combine different ways to be able to confirm what was going on, because you can't just rest on a test that has such high false positives. So just continuing on here with this specific assay and trying to build it out so that you can make it more reliable and eventually hopefully a singular test for diagnosis.
That's it. And you bring up a good thing that, we just covered all the pre-analytical conditions. But then now if the sample's at the lab, what do you do with it? Because with whole blood, it's pretty clear like what cell types you need to look for and things like that. But with these types of non-standard matrices, building a panel, defining your cell types of interest, finding the right reagents can be a little bit more of a crapshoot, right?
Hmm.
In that you're not just looking for your standard TBMNK lineage. Here, you might need to look at epithelial cells. You might need to apparently gate out SECs. And then the question is, are there reagents actually available to tag all these things?
Yeah, and the most obvious corollary I just jumped into my mind as you were talking about that is whenever we're challenged with a different species type than is the normal, right? We've gotten obviously cyno, we've gotten mouse. Those generally have a lot of support for antibodies, but then you start getting analysis of dog. There are fewer markers there that are available, fewer antibodies that are available. Gotten pig before, llama, so on and so forth. These all of a sudden it just becomes that much more difficult to be able to build out, and it's possible. I mean, like I said, all these things we've done, and then here it really is you're going into that different sample type, and so they're looking at these epithelial cells, and they got markers for those that were reliable, and then as a counterpoint to that, having the markers for the immune system, which thankfully, at least, they're not changing, right? Your T cells, they did talk about the different types of macrophages that are present. And so here specifically, they were looking at CD206, which is present in this sample type. However, in other macrophages are more a hallmark of your non-classical, and so wouldn't be an umbrella analysis of macrophages, for example.
That's it, right? So sometimes cell populations are there, but may look different or be taking on a different function because of where you're getting the sample from. Again, sputum is one thing, and then tissue is another. So tissue resident macrophages would likely look quite different than sputum macrophages potentially.
Mm-hmm.
So being able to gate based on those differences, I found it interesting that they use the macrophage number as a quality control as well.
Yeah, it did. So what was it? 5 in 10,000 cells as the cutoff, and so if it was below that, they didn't feel like it was reliable, and had thrown it out. And so, yeah, again, that's where they were using the anti CD206 to be able to identify, and just talks to what they felt was needed to be able to get a conformational diagnosis there.
That's it. And it just speaks to, again, just the challenges and the amount of expertise you have to be able to do one of these non-traditional matrices, that you really have to understand what is supposed to be in that type of sample and how it could change and what that change means in terms of the disease state. So yeah,
So
interesting stuff.
overall, I would agree with your assessment. I really enjoy reading a paper that tells a story, and I think this paper really tells a story in how they went about everything. Not every paper is going to have the final finished product, but this is the panel, this is the diagnostic test. You've got to have the iterations to get to that final step, and so I think they really did a great job of walking you through what they had done to get to where they did.
Yeah. Now, in terms of decision making for clinical trials, if you had to analyze a sputum sample, would you try to limit that decision making to just the presence or absence, or would you feel comfortable making quantitative assessments based on what we saw from this paper, at least?
I don't know that there really is quantitative assessment here, because a lot of the populations you're talking about high, medium, low, not plus or minus. Obviously, that's something that we deal with a lot in blood samples too. I don't want to say that we don't, but here I still feel like this needs more, right? Like you can't just rely on this test alone. I know that's not necessarily the question that you're asking, but what are your thoughts on it?
I agree with that, right? That I think you could make a very binary decision, like is something present or not present, or is something above a threshold that you define as high. But I would struggle to make real quantitative comparisons for this type of matrix.
I think, though, that the hope would be the TCPP fluorescence. That specifically, if you do that prior to fixation, do you get the answer that you're looking for? Like, I would have really liked to see them kind of titrate between healthy and cancer to be able to verify that they were able to see that specific bias there, where they didn't get that. That was going to be the driver for this paper. Like, in reading this, my feeling was they were hoping that was going to be the answer, right? And they'd see this huge bias between the cancer versus the high risk, and it was predictive. But unfortunately, it just didn't happen. And I think it may just be the fixation, which they owned up to right at the end. And so be curious to see if there were next steps that would follow up to that.
I bet there are, because of course this paper came out in 2022.
Yeah.
And the company that initially was developing this test is still around, and has a test on the market. It's marketed as a lab developed test, not a completed IVD assay.
Right.
But hopefully they resolved this issue and found something, and they just didn't, of course, publish the final one.
I'm going to guess they did if they're selling it as a test, but no, I know what you mean.
Yep. So exciting times, and as you say, always fun to talk about a new matrix and something a little bit off the beaten path here. So appreciate that.
Yeah, absolutely. All right. So what do you got for us next?
Next up is feedback request. So what we are looking for one of our upcoming episodes is, what is your favorite or least favorite control strategy in clinical trials?
That sounds fun.
For QC people, it is quite fun. For others, maybe, I don't know, it's a little bit dry. But I like to think about isotypes or FMOs or Strec or versacells or just healthy donor PBMCs. What is either easiest or best? And it's a little bit of a context dependent question, but I definitely have my preferences in terms of what is well behaved over the long term there. So
definitely context dependent, right? I do remember starting out in Flow, that was the huge debate. Like, everything had to have an isotype control. And at that time, at least from my experience, it really wasn't as much in the way of FMOs. And I feel like that's shifted significantly, right? And so curious if other people feel the same way, but many assays, most assays are built on FMOs as opposed to isotypes. There are specific instances you see the isotypes. But anyways, this is next episode, so I don't want to go diving into it too far.
Yeah, and hopefully it generates some good discussions with the fans. So looking forward to that.
Mm-hmm.
All right. And then last is just wrapping up. So in terms of our end of episode banter here, it being August, what small ritual or item symbolizes back to school the most in your memory?
So I got a couple of different things here. You'll have to apologize for my scattered nature on this. There's the two parts to my life, or like with the kids, and then when I was younger, right? And I'll just start by moving backwards. And with the kids, what we would always do was go to Dairy Queen right after the first day of school, and it was always a lot of fun. Get like a Blizzard and chill out. And then the same thing for the last day of school. We would end it by going there. But as far as for myself, now we're going back 100 million years. Obviously love those pens, like the 4-color pens that had like the blue and the white, and you can click it and change the colors. But I mean, you're just going to roll your eyes at me on this one. Hopefully you don't think too much less of me, but using those pens where you could take out the guts and then use it to make spitballs.
Oh yeah.
That was a lot of fun. So there you go.
I mean, with the 4 different things, you could shoot 4 spitballs. So yeah, this is an exciting time.
Yeah. I don't know. I never was able to get one of those to do that. This was just like the Bic that it's a tube, right? You put the ink down the tube and you chew up a little piece of paper and then you shoot it out. So
okay, so you're going with the blow dart method.
Yes,
I was thinking of using it as like a spring-loaded attachment there.
No, get the blow dart. And if you had to go towards less crazy methods like that. The less memories, definitely those blue binders, right? Like almost had that denim on the outside. I love those. And
with the zipper.
no, not with the
Okay.
zipper.
Okay.
Not
All
with the
right.
zipper. Just the standard one.
Okay.
Maybe the less expensive one. And then obviously the marble notebooks. So now that I picked everything, why don't you go?
I was going to say the trapper keeper, right? So at the particular moment in the 80s or 90s or whatever that I was going to elementary school, definitely the trapper keeper and how cool your pencil case was were big things.
Mhm.
So we did have the notebooks with the zippers around them. I hope they weren't expensive. I don't know, but that was critical for school success there to make sure that all your stuff stayed in one spot, and the amount that you could cram in there was absolutely ridiculous as well. So,
Oh yeah, they were your friend until you dropped it and you hadn't had it zipped up, and then all the papers go everywhere. Did you say that you were in elementary school in the 90s?
it's best not to think about it, Adam. I know it's not 100 million years ago, but
Oh my goodness. Oh.
All right. Well, excellent episode. Thanks to all of our listeners for being with us for this. Looking forward to your feedback. For next episode in September, we are planning to schedule the "What Happens If You Go with the Flow Too Much" episode. So looking forward to that.
That sounds like a water park episode. But anyway, I digress. Yeah, thank you, everybody. Really appreciate you joining us. And please share feedback if you liked anything, you didn't like anything. Always love to hear from you.